In our last video, we did 3 examples of Hardy-Weinberg equilibrium and took you through the whole process, but it really is worth practicing again. If you feel confident, you know how to do it exactly, you can skip ahead and just go to the PowerPoint part of this video, but I think it's worthwhile, especially if you're having a little bit of trouble. So I've got 2 examples we're going to work through. The first one, we go into a population and we sample 1,000 individuals. We find 360 individuals are big A, 480 are heterozygotes, and 160 are homozygous recessive, or little a. So your goal is to take these genotype counts, turn them into allele frequencies. Those are your P and your Q values. Take the P and the Q values and plug them into the Hardy-Weinberg equation, and then determine whether or not this population is at Hardy-Weinberg equilibrium. Pause the video now, and then we'll go through the results together. So this is what you should have ended up with. I do 2 times 360 because there are 2 copies of the big A allele in these 360 individuals. I add in 480 because the heterozygotes carry 1 copy. I get the total number of big A alleles. I do the same and get the total number of little a alleles. I then divide by the total, which in this case is 2,000 alleles. Though that number comes from 1,200 plus 800, and I get 0.6 and 0.4. Okay, that's my P value, my Q value. I plug them into this Hardy-Weinberg equation, do a little bit of math, and I end up with predicted genotype frequencies for the next generation. So remember, this first number is a prediction for P squared, which is a prediction for the homozygous dominant, 2PQ, a prediction for the heterozygotes, and then Q squared, a prediction for the homozygous recessive. Okay? Now, at this point, I can check to see whether my predictions match my original observations. And in this case, they do. 36% or 360 out of 1,000. 48%, 480 out of 1,000, and so on. So for example number 1, we are in Hardy-Weinberg equilibrium. So now let's take a look at example number 2. Again, the same thing. Go through the same process, get it all into the Hardy-Weinberg equation, and then determine whether or not this population is in Hardy-Weinberg equilibrium. Pause the video now and come back and check your work. So this is the end result. Same exact process, right? So I won't take you through all the details because we just did it. But look at the results. We predict a little over 30% of the population should be homozygous dominant, but it's actually 48%. We're predicting a little under 50% should be heterozygous, but we only have 16%. And then, of course, this number is out of balance also. So this population is not in Hardy-Weinberg equilibrium. Okay, so for the rest of the discussion, let's talk about the things that cause populations to not be in Hardy-Weinberg equilibrium. So that equation, that prediction, becomes in its essence a null hypothesis, which is if there are no evolutionary forces acting on a population, everything for that allele should balance. Now it's important to remember when we're doing Hardy-Weinberg equilibrium, we're only looking at one gene, the 2 alleles for that one gene. And other genes could be changing and evolving, but other alleles might be in equilibrium and stay exactly the same generation after generation. So this null hypothesis is a prediction for what the next generation will be, and it also tells us if there's any evolutionary pressure that's acting on that gene. So there are 5 things that can impact Hardy-Weinberg equilibrium and throw it out of balance or violate our null hypothesis. Okay, so here they are. Now, 3 of these you're already very familiar with. Natural selection, genetic drift, which is small population size. That's when genetic drift is in effect. So in fact, so hopefully you recognize why, but if not, we'll get to that in a later chapter. But we're going to replace small population with genetic drift. And then, of course, new mutations. If those things are happening to an allele, then we will see imbalances. They may be slight or they might be large. There are 2 other things that are added. Now, some people will put these 2 other things under the umbrella of natural selection, but for our purposes, we're going to separate them. So non-random mating, that means if you are choosing a mate based on the gene that we are investigating, and we'll look at examples of that in a later chapter. And then migration. If we have individuals migrating from a different population with different allele frequencies, that can throw our balance off. Now, it's important to recognize that there are many, many different balance points for the Hardy-Weinberg equilibrium. This is a three-dimensional representation of all of the possible outcomes of the Hardy-Weinberg equilibrium. And it looks a little bit like a half pipe, right? A skating half pipe. So if you threw a marble into a skating half pipe, and if it was perfectly balanced on the bottom, there are multiple places. Anywhere along the bottom of that trough, you could have a balance point. And the same thing with Hardy-Weinberg. You can balance with 50-50, you can balance with 20-80, you can balance with 30-70, anything as long as all of these forces are not acting on the population. You can reach balance at many, many different points. Okay, so that's important to remember. So let's look at some of these things. Mutation on Hardy-Weinberg, we're just going to treat very, very quickly because mutation is random and unpredictable. And so we can have all sorts of different new alleles, but that complicates things. If we have a new mutation and suddenly we have 3 alleles instead of 2 alleles, the Hardy-Weinberg equation becomes more complex than we want to deal with for this class. Okay? But I do want to generally talk about it because new mutations are happening all the time. Many of those mutations are negative, right, or deleterious. They have a negative impact on the individual that carries them. But then we have forces that try to remove those negative mutations from the population. And by remove, I mean individuals carrying mutations don't do as well, they don't have as many offspring, and so that mutation is underrepresented in the next generation. And if that continues, eventually we will remove those negative mutations completely from the population. So deleterious alleles or negative mutations reach a balance that depends on 2 factors. Number 1 is the rate of mutation, or how many mutations occur each generation. And remember, that can vary. In some organisms, mutation rates are high, and there's lots of variability introduced each generation. And in some populations, mutation rate is low. But there's always going to be some level that's bringing in new mutations, and some fairly significant, maybe small, but significant amount of those new mutations are going to be detrimental. And so we have mutation rate bringing in some amount of bad mutations, and then natural selection working to remove those negative selections. And so the balance point depends on those 2 forces. Mutation bringing in negative things, and natural selection working to remove those negative things. Now there are neutral things and occasionally a rare positive or beneficial thing being brought in by mutation too. But for now, we're only going to look at these deleterious alleles in that general sense. So migration is another thing that can greatly impact populations. So let's redefine population. And we talked a little bit about this in our very first lecture, but let's just clarify it. A population is any subgroup of a species. So an entire species can be a population, but we can also divide species into smaller populations. And there are numerous names that we use for this. In the human population, human species, we use sometimes race or ethnicity to define a subpopulation. In other groups, we might call them a breed, right? So for dogs, we have different breeds that represent different subpopulations of the dog species. Plants, we might call them cultivars. In viruses or bacteria, we may call them strains, but those represent different subpopulations of a much larger population. And so it gives us a little bit more detailed definition of population. Populations can be entire species, but we can also subdivide them, and those subdivisions are based on some sort of genetic separation. So here we have 2 subpopulations in a species, one on the mainland with a different allele frequency than one on the island. And if we have migration one way, maybe there's a current or common weather pattern, winds that make migration only occur in one way. If that's occurring, that is going to throw off the Hardy-Weinberg equilibrium in this population on the island. Now, eventually, if enough of that occurs, it will reach the same equilibrium that we see on the mainland. But in the time being, until it reaches that point, we are going to have a little of an imbalance in the Hardy-Weinberg equilibrium. And so if we put in our numbers and saw that imbalance, we would say, oh, there's something going on here. And so when the Hardy-Weinberg equilibrium, when our predictions don't match our original observations, we know that there's an evolutionary force acting on a population. Now, it's tricky sometimes to figure out what that force is. It might be natural selection, it might be genetic drift, it might be mutation, it could be migration like we have here in this example, or it could be a non-random mating. Okay, so let's look at non-random mating. So in non-random mating, there are basically two general types and lots of degrees in the middle. So we have assortative mating is when like individuals mate with other like individuals. We have disassortative mating when mates prefer to choose an individual who is different from them. So assortative mating is also known as inbreeding, when individuals mate with individuals that are genetically similar to themselves. And inbreeding has some negative consequences. The most extreme form of inbreeding is self-fertilization, right? If you breed with yourself. Now, not all species can do that. Most animals cannot. There are a few exceptions to that rule. But many plants can self-fertilize. And so if we have self-fertilization, you're mating with yourself. That's the ultimate genetic similarity. If there is too much inbreeding, we get something called inbreeding depression. Now, a lot of people think, oh, if you mate with other individuals, there are going to be more mutations, and that's bad. It's not that there are more mutations. But here's the reason why inbreeding can cause problems. Every individual, on average, carries 2 or 3 deleterious alleles. And in fact, there's a really good chance that you carry a fatal allele. Now, hopefully that doesn't concern you too much, because almost everyone does. However, you are carrying a fatal allele that is recessive. And it means you got one bad copy from one parent and a good copy from another one. And because it's a recessive fatal allele, it's not a big deal, because you have 20,000 some odd genes in your genome. There's a very, very low chance that your mate carries that same recessive allele, unless that mate is closely related to you. And then the odds that those rare recessive alleles that are fatal are the same in you and your mate, those odds jump up quite a bit. And especially if it happens generation after generation. So inbreeding depression, the reason that it's a bad thing is because of the pairing of those rare recessive, lethal, or at least extremely detrimental alleles. And that's called inbreeding depression. Now, in many species, there are instances of disassortative mating. One prime example of that is MHC in mice. So we've got an experiment here with the birds where if we put a female bird into an experiment and give her the choice of a male bird that looks different than her and a male bird that looks much the same as her, she will more frequently choose the male that looks different. That's just a mutation that popped up in that population. It was a beneficial thing. Because it reduces the chances of inbreeding depression. If females on average choose mates that are different than themselves, then they're going to pick mates that are more genetic. Their offspring are going to be more genetically diverse. MHC in mice is another one. So MHC stands for major histocompatibility complex. In mammals, that's an important determinant of your immune system. And the more diverse those MHC genes are, the more diverse and better your immune system will be. And so if we do the same experiment where we put a female mouse into a chamber and let her choose a male that has the same MHC complexes or different MHC complexes, she will more often choose the mate with different MHC complexes. Now, how do they know? How are mice determining that? Well, almost certainly it's a chemical cue, and I think there's even been some research finding out exactly how they do this. But it's one of the ways that mice assess mates. There's probably something similar going on in many mammal species, maybe even in humans a little bit subconsciously. How do we assess a mate? Do we do it purely on visual things? Do we do it on other communications like, verbal communications or physical cues? Is there some sort of a pheromonal thing, a chemical cue that humans are using? And there is some evidence for that. But certainly in other animals, that's much, much more pronounced. This is the reason dogs go around sniffing other dogs' butts, right? To put it kind of, I guess it's a little bit crude, but not too crudely, right? They go around, they sniff their butts. They're assessing chemical cues. Is that individual fertile? Is that individual sick? perhaps even other ones. Is this individual the same as me, is different than me? And they're doing this all because these random mutations for these behaviors have popped up. If they're good, they're kept around. If they're an advantage to the population.